Hello everyone. So I welcome you back to this new series where I am going to discuss the topical past papers for your IGCSE's biology. And here we are beginning with topic number two. And the title of the topic number two goes as the organization of the organism. So we are about to see that how come the examiner is going to give you the questions in the exam from this topic. So if we just look on to question number one, he asks us that we have got this hemoglobin, and this hemoglobin is a protein, and that is made inside the developing red blood cells, and that too in the bone marrow. So if you were to recall, we know it very well that these red blood cells, they are the specialized cells, and they appear as a biconcave discs. And in order for the red blood cells to be binding with the oxygen gas, so they have got the packaging of a protein in them, and that is hemoglobin with us. Now, if we say that the red blood cells they have the hemoglobin protein, so the idea is very clear that if I have to make this protein, so I need to combine the amino acids together, and they would make the polypeptide chains, and that would further fold up to be making this hemoglobin protein with us. So he has given you three parts, and for the first A part, he says as or he asks as that you are required to state him the function of the hemoglobin. So the protein hemoglobin within the red blood cells, you could say as that it would transport the oxygen gas. You can also write an alternative point for that, that the role of hemoglobin is not only the transport, but it would also be binding with the oxygen gas within the alveoli of our lungs. In the second part, he asks us that name the small molecules that are combined to make the hemoglobin. So, owing to the fact that this hemoglobin is a protein, so I am required to be linking the amino acids together. And as I mentioned earlier, we would make the polypeptide chains, and finally, we would end up making this hemoglobin protein. Further, he says as that name the mineral iron which is provided in the diet, which is needed to make the hemoglobin. So, since we have to focus on the word mineral iron, so the mineral iron that is available within the hemoglobin, this is iron with us. Moving on to question number two, and they have given you figure 1.1, and it shows you some cells that are actually lying in the trachea. So if I just simply highlight the components within these cells, so here you see that the examiner has given you the cell membrane of this ciliated cell. Here we have the nucleus. For sure there would be presence of the cytoplasm here, and here we have got small finger-like projections, and they are the cilia with us. So this cell, and this cell, so they are the ciliated cells. But we have got another cell that is given here in the diagram, and this is the goblet cells. And these goblet cells, they are the excretory cells, owing to the fact that they would release a sticky substance out of them, and that is the mucus with us. Now, he has asked very simple things from this section, and he says is that for the figure 1.1 that we have given you, you are required to tell him the function of the nucleus and the cell membrane, and it should be in term of description. So for the nucleus, you have got multiple approaches to be keeping it in your mind that the nucleus would control the activities of the cells. If you don't want to write it down, then we also know that this nucleus that we have, so it also contains the genetic information in it. And even if you forget this point, so you know that at some point our cells they would divide by mitosis to be making two daughter cells. So this third function of the nucleus is that it controls the cell division. So since the marks given are four, so you are required to be writing any 2 points out of these three, which I have written it for you. If we were to be talking about the cell membrane, so if I just make the diagram, here we have the cell, and for the cell we have the cell membrane. So we know that this cell membrane is partially permeable or selectively permeable membrane. So only the useful materials they would be allowed to entering into these cells. And at the same time, you know that we have got this cytoplasmic content within a cell that we would like to be keeping it intact within the cells, and we would not allow it to leak out of the cells. So if I have to control the entry and exit of substances, and if I have to keep the cytoplasm intact or the cellular content like the mitochondria is intact, so I would be in the need of this cell membrane with me. So for the cell membrane, if you are required to be writing the functions, so you could say that it would separate the content of the cells from the external environment, or you could also say that it can control the movement of substances, and you are required to be giving your examiner an example of the material that whose movement is controlled. And as also you if you forget about oxygen, so you could name some ion like the calcium ion or some chloride ion or stuff like that. So this would be able to move in and out of cell with the help of the cell membrane. Now he asks in the B part is that the cells which they are showing you in figure 1.1, they say that they would form a tissue. And for 1 mark, he is asking you that you are supposed to be defining the term tissue. So for the tissue, what we need to know is that the tissue is always a group of cells. And these group of cells, when they work together, so they would be performing a specific role. So for this much, you would get 1 mark. Now, if I were to further proceed to this question number 3 that we have got over here, so they say that we are showing you figure 1.1, and this is a cell from the region B of the leaf shown in figure 1.1. So some part of this question is missing. For sure, there would be a leaf with us, and from the leaf, he has given you the diagram of this cell. So if I just quickly tell you that what components do we have with us. So if you focus on the structure F, so this structure F right here, so this is indicating the nucleus. The G that we have over here, so this is the cell membrane. The H that we have got is, if you just clearly could see, it is going to this kind of a dot here. So these are the ribosomes with us. If I further proceed to this J part that is here, so this is a cell wall. And if I talk about the K, so this is the large vacuole of this plant cell. And L, that latter which they have given us, so they are indicating the chloroplast. If I were to give you a tip from the paper point of view, in your exam, the chloroplast would be given like this, and it would be shaded with the black marking. The other way around is that they would show you some stuff like that, and then they would give you the presentation of the membranes like this. So this would confirm that this is the chloroplast with us. Now, if we further see, they have given you the box. They ask is they inquire from you is that you are required to be using the letters from figure 1.1, and you see that we are given multiple letters here, and then we are required to be completing the table. What we actually have to keep in our mind is that we are supposed to be writing one letter in each of the box which they have given us that actually applies to the function which they are rendering. And if we just simply just solve this up, so here they say is that the function they have given is that they control the movement of the substances into and out of the cell. So I said this is the cell membrane, and the letter that we are given here, this is G with us, okay? And then they say that something exerts a pressure to help maintain the shape of the cells. And we know that the maintenance of the shape of the cells is basically dependent on its large vacuole and the way it would exert the pressure onto the cell wall. So if they are saying that something exerts the pressure to maintain the shape of the cell, so this would be the large vacuole, and the letter for the large vacuole given here is K with us. The thing doesn't end here. They ask is that produces the sugar using the light as a source of energy. So what we know is that the production of the sugar like the glucose, this happens within the chloroplast. So if you just see the letter L that we are given over here, so this is a chloroplast with us, and then they say is that withstands the internal pressure of the cell. So the turgor pressure is applied by this large vacuole, and if someone has to bear this turgor pressure, so the cell wall would be contributing the role in it, and the letter that we are given over here, this is J with us. And they say control all the activities of the cell. So you all know that the cell's activities are controlled by the nucleus. And the letter that we are have to be writing over here. So this is F with us because this is the nucleus that we have got. I hope all of this is making sense to you. And if we just go on to word question number 4, they say we are showing you this figure 1.1. And this figure 1.1, this shows you an animal cell and a plant cell. And there you could say image, it has been taken with the help of a light microscope. Now, why this is an animal cell? Because we do not have any of the cell wall being made in its structure. And if I talk about this cell which they have given us, so this is a plant cell owing to the fact that we see the cell walls and then we see the large vacuole and then we also see the chloroplast over here. Now, if we just look on toward the question which they're asking is that we have given you table 1.1 and it shows some structural features of the animal cell and the plant cell. They say you are required to be completing the table, and they have given you this table 1.1, and they say is that you have to be finishing the row for the nucleus, and then along with that, you are required to add three structural features that are shown here in figure 1.1, and you are required to be indicating that whether they are present or they are absent in the animal cell and the plant cell. So if we just simply go on toward the table, the first row we are completing it for the nucleus because the cell wall has already been done. So here is a structural feature, then are the concerned cells with us. So if we talk about the nucleus, so you see that the nucleus is available within the animal cells, and this is also available over here in the plant cell. So we would put the tick in both of these. Now, for the other things that we are required to be writing here, so that are actually they say that it should be there in the diagram. So you see is we have got the cytoplasm, isn't it? And then you see that here we have got some small tiny dots. So they are the ribosomes. You can also be telling about the large permanent vacuole, and you can also be writing about the chloroplast. But depending upon the animal and the plant cell, you are required to be putting a tick or a cross. So I have included cytoplasm, and you know this is available both in the animal and the plant cell. The other feature that I have taken from the diagram of the plant cell, these are the ribosomes. As you see in the animal cell, we don't see any of the ribosomes, but in the plant cell, we see them up. So we would put a tick in both the cells sections because the ribosomes is there both in the animal and the plant cell. And if I talk about the large vacuole, so I would put it across for the animal cell, but I would put a tick for the plant cell. Now, if you were to be writing about the chloroplast, so it's your choice. Like instead of writing this, you can also be writing the chloroplast here. Moving onward, this is one of the very typically asking question from the specialized cells from the plant, and that is the root with us. They say is that this figure 5.1, this is an image of the germinating seed, and this is showing you the growing root. Now, if you were to see, here we have got this germinating seed with us, and here is the root of this germinating seed. And out of the root, you see is that we see these tiny projections coming out that are indicated with the J letter. So they are the root hair cells. Now, if I quickly recall what the root hair cells they have in them, there would be the availability of these cell wall in them. And if I come immediately below the cell wall, so there would be the presence of the membranes, and these membranes, they are the cell membrane. And one of the good feature about the cell membranes of the root hair cell is that there would be the availability of some specialized transport proteins, and they are the carrier proteins. And these carrier proteins, they are important for the active transport of the ions, or they are important for the for facilitating the movement of the ions. Now, if we go like at the center of this cell, so here we have got the large vacuole coming with us, and then there would be the availability of many mitochondria here within their cytoplasm. So here they have given you total of 5 marks, and they want you to be describing and explaining that how the structure that is seen at J, this is adapted for their function. Now, if you were to recall from your class lectures, what we know is that these root hair cells, they have the major role in absorbing the water and the ions, and for that they need to be adapted. Now, I have written the first point, and I would tell my examiner that J are the root hair cells. And if I were to be talking about other, you could say, structures that would adapt them for the function. So you have to be talking about these carrier proteins for the movement of ions. Then you have to be talking about the abundant mitochondria that would release energy by the aerobic respiration for the active transport of the ions. And then you would say that these root hair cells, they have large surface area to the volume ratio that would bring about the efficient absorption of the water and ions. You could also say that these root hair cells, they are long, and that if they are long, so it would accommodate them in penetrating in between the soil particles. The other point is that these root hair cells, they are very thin, and when they are thin, so it would reduce the distance for the diffusion process. So if I were to simply like be telling you these points, so we would say that they have the large surface area to the volume ratio, and that would be bringing about the efficient absorption. Then I have said that they are thin and long. so it would allow them to spread easily around the soil particles. Then I have written that these root hair cells, they have abundant mitochondria that would allow them to release energy and that would be utilized for the active transport of ions. And then these root hair cells, they are also thin. And when they are thin, so for sure the distance for the diffusion of the substances would be reduced. And then you must never forget, in terms of the root hair cells, that they have got the carrier proteins. The issue with the ions is that these ions, they cannot directly cross the cell membranes. They cannot cross it. So if I want the ions to be passing into my cells, so there is the need of the carrier proteins. And these carrier proteins, they would facilitate the movement of the ions into the cells. So here you would score a total of 5 marks. Now, if I come on to this question number 6 that we have here. So this figure 2.1, and this is a drawing, and it is made from the electron micrograph from a cell, and that cell is a lymphocyte. And a good thing about the lymphocyte is that this would be making the antibodies. Now you know as that there is availability of the pathogens in our external environment and they can enter in our bodies. So my body has got these lymphocytes and the phagocytes, and the lymphocytes would produce antibodies, and antibodies are proteins with us. And these antibodies, they would provide the protection against the pathogens. So if I just simply try to be telling you that what this diagram is indicating, so this A part that we have with us, so this is cell membrane. And if I talk about the B, which is indicated over here, so this is the nucleus with me. If I were to talk about the C structures, so you see that they are granular, they are spherical shape. So you are required to be calling them as a ribosomes, and the role of the ribosomes, you know, is that they would make the proteins. If I talk about D, so this is cytoplasm. And if we talk about E, so this double membrane organelle that we see over here, so this is mitochondria, and the F, which they have indicated you here, so you can call them as vacuoles. And this interconnected network of the membranes that have got these granular structures on them, so collectively we call this as the rough endoplasmic reticulum. Now, let us see what the examiner has to ask from here. They say that this table 2.1, which they have given you below, so this contains the statement about the structures that are visible in this figure 2.1. And they ask you that you have to be completing the table 2.1 and you are required to be naming the structures, and then you are also required to be identifying the letters that would correspond to the structures which they have given you. And for your ease, what the examiner has done, he has solved the first part. So here they say the function is absorption of amino acid that are used in making the antibodies. And the name of the structure which they have given is a cell membrane. And the letter which the examiner himself is writing, so this is A with us. Now we have to be cracking the other things. They say that what stores the genetic information, and we know this is the nucleus. And the letter I have already indicated, this is B with us. They say that provides energy for making antibodies. Now, since we need to make antibodies, that would require the energy. Energy is always released by the mitochondria with the help of the aerobic respiration. And the letter that we have got for the mitochondria, this is E with us. Now, here they have asked you that what is the site of the production of antibodies, and what we know is that these antibodies, they are the proteins, and proteins are always synthesized by the ribosomes, or they are made on the rough endoplasmic reticulum. So the choice is with you. Either you have to write ribosomes or you have to write RER, but the choice of the letter would change. If you were writing ribosomes here, so the letter that you are required to be giving is C. And if you were writing the rough endoplasmic reticulum in that column, so you need to be giving him the letter G that we have got over here. And then they say is that what would be transporting the antibody molecules out of the cells so it could be released into the blood. Now the idea is that after these ribosomes that we have here, they have made the antibody. So this antibody has to be released out of the cells, and for that we are in the need of the vesicles or the vacuoles, and these vesicles or the vacuoles, they would pack the antibodies and then they would fuse with the cell membrane so that the entire body molecule could be released out from here. So that is why I have written the answer as vesicles or vacuoles, and the letter that I have given him is F with me. Now, if we come on to word question number 7, they say that we have got this kingdom fungi with us, and this contains a great diversity of the organisms. And among the funguses, we have got the yeast, then we have the molds with us, and then we have got the mushrooms with us. Okay, and then they ask us that, like the plants and the fungi, they also contain the nuclei and the mitochondria. So we know that the plants they have got these nucleus and the mitochondria. So the fungus would be containing these structures as well. Now for the 2 marks, the examiner is saying that you are required to be stating two characteristics of the fungi that are used to distinguish them from the plants. Now we know it very well that the plants they have the chloroplast, but the funguses they never have the chloroplast. The plants they are autotrophs, they make their own food, but the funguses they are heterotrophs. Or in other words, we could say is that they are saprophytes because they feed on the dead organic matter. If we were to be talking about the plants, so they have the large vacuole in them. The funguses they do not have them. The other striking thing is that the funguses and the plants, both of them, they have the cell wall. But the difference is that the plant cell wall is made up of the cellulose and the fungus's cell wall, this is made up of the chitin. So I have told you many points and you are required to be writing any two. So you could say they are heterotrophs. If you don't want to write it down, then you see I have written an alternative point and you would say that they do not possess the chloroplast. And then you would say that there is no central vacuole that we have got in the fungus, and you can also write about the difference that we have in the composition of their cell wall. Now, moving onward to question number 8, they ask is that glycogen is a storage carbohydrate in the animals and the glycogen is made from the glucose. What we know is that in our body we have got this organ, and this organ is the liver with us. And this liver is connected to the small intestines with the help of the vein, and the vein that we have got at this point, this is termed as the hepatic portal vein. The usual thing is that we human beings eat more glucose than is required by my body. So once the excessive glucose comes into the liver from the small intestines, the pancreas would detect it. And when the pancreas would detect it, so they would send a hormone toward the liver and the muscle cells. And that hormone is the insulin. The insulin would activate the enzymes in the liver, and that would convert the excess glucose into the glycogen. Now, for the A first part, they say that the cells that convert the glucose to the glycogen, they have many mitochondria. Now, once the examiner says it has many mitochondria, so this would indicate as that the conversion of the glucose to the glycogen, this is an energy intensive process. A lot of energy has to be produced for this conversion to take place. And we know that this would require the mitochondria because the mitochondria they release energy by the respiration, and that would help the conversion of the glucose to the glycogen. So for the 2 marks, the examiner is saying that they want you to be suggesting him that why these cells contain many mitochondria. So you would tell him that the conversion of the glucose to the glycogen, this requires energy, and for the energy production, there has to be presence of many mitochondria. So they would carry out their aerobic respiration and the energy would be released, and that would convert the glucose into the glycogen. If I were to further move to the second part, and the marks given are 1, so here they ask is that state the type of the biological molecule that catalyzes the reactions. Now, since the keyword used here is catalyzes, so you know it very well that the chemical reactions are always catalyzed by the enzymes. So the glycogen to the glucose requires enzymes, and these enzymes they are actually activated by this hormone with us, and this is insulin with me. Now, if we were to be moving on toward this question number 9, and they say that I am showing you, the examiner is showing you figure 6.1, and he has shown you some cells, and these cells they are coming from the shoot tips from an onion plant, and the genus is Allium, and the species is Kappa. Now you see that this is a shoot tip. So what my expectation is that these cells for sure they would have in them this cell wall, and this is confirming that okay fine, this is a section from the shoot tip. Now they ask us that state the evidence which is visible in figure 6.1 that identifies the cell of the say kappa as a plant cell. So you see that we clearly see the cell wall being present here. And then they ask us that the cell A is dividing by mitosis. And by the mitosis, what I mean is that at the top, I have got the parent cell. And this parent cell, whenever this is going into the mitosis, so this would lead toward the formation of the two daughter cells. And you see that this process where the parent cell divides to be forming the two daughter cells, this again requires energy because in this parent cell, I am required to be duplicating the number of chromosomes. And again, the energy would be required to be moving the chromosomes equally into the two daughter cells. They say that state the role of mitosis in the shoot tip. Now you see that the role of the mitosis is simply that we lead toward the formation of new daughter cells. So if the new daughter cells are formed, so they are equivalent to be bringing about the growth in the shoot tip. Or else you could say as that it would be producing the new cells. But if you don't want to write it down, you could also say that this mitosis occurs and that brings about the elongation of the shoot for the plant cell. Now, here they ask is that the area labeled M is a mitochondria. So if I move you back to the figure which they have given us, so here we have got this structure and this is M and they say that this is mitochondria. And what he asks is that you are supposed to explain why the mitochondria have an important role in dividing cells. Now, as I have said and mentioned earlier, that if I have this parent cell and further if I have got the formation of two daughter cells, so this parent cell needs the energy from the mitochondria to be duplicating the DNA or to be duplicating the chromosomes. And for sure, I have to move these chromosomes equally into the new formed daughter cells. So that again would be requiring the energy process. So for 3 marks, how you are required to be answering is that the cell division that occurs by the mitosis, this requires energy. Here you will get 1 mark. And then you would say that hence these cells contain mitochondria. Why? So that they should release energy by the process of respiration. And once we have released energy, so you have to link it to some process that is happening during the mitosis process. So you would say that this energy is used for either moving the chromosomes from the parent cells to the daughter cells, or you could also say that the energy released by the mitochondria is also used to be making the new chromosomes. So this is a separation that I have given you. That either you could write this point or you could write this point as a substitute. If we come on toward this question number 10, so they say that here we show you figure 6.1, and this is a photomicrograph of the part of a cell from the pancreas, and that too is producing the enzymes that are released into the small intestines. You may have learned in the chapter of nutrition in humans that if we have got this liver, so, and then moving onward, here we have got this gallbladder. And then here there would be the presence of the pancreas. And then all of them, they would be connected to the first part of the small intestines, and that is the duodenum with us. So whenever the digestive enzymes are required, so this pancreas would produce a pancreatic juice, and that pancreatic juice, it would come into the duodenum of the small intestine and it would have the enzymes in it. Now, if we just look on toward the A first part, they say structure Q is the part of the nucleus of the cell. So here you see they have given you the Q here, and this is showing you some part of the nucleus. So this means that this part right here, this is indicating the nucleoplasm of the nucleus. And if you were to just look here, like here is a sausage-shaped organelle, where we have got the presence of the membranes like this. So this is a point that identifies this part labeled R as the mitochondria. And if we just look on toward the letter P, so here we have got an extended network of the membranes. And this has got some kind of a rough appearance or the granular appearance. So this P that we have with us, so you can call this as ribosomes or the rough endoplasmic reticulum. Now they ask for one mark is that you are required to be stating one function of the nucleus, and you would say that it controls activities of the cells, or either you could say that it contains the genetic information, or you could also say that it controls the cell division. So any one point from these three which I have written for you. They ask further is that you are required to state the name of the structures that are labeled as P and R. So you see, as I mentioned earlier, this P you can write them as rough endoplasmic reticulum, or you can call them as the ribosomes. And if we talk about R, so this R that is given over here, so this would be mitochondria with us. And by that, you would score 2 marks for this. Let us come to this question number 11 with us. They say that figure 1.1, which we have given you here, this shows a spongy mesophyll cell from the leaf of the plant. Now we know that the leaf of the plant is an organ. It has a cuticle, then it has got upper epidermis, the lower epidermis, then we have the palisade, and we have got the spongy mesophyll cells. So they are showing you the diagram of the spongy mesophyll cell. They say the arrow shows the net direction of the movement of the carbon dioxide molecule during the daylight. So you see here these arrows, they are indicating that the carbon dioxide has come from the external environment into the air spaces that we have in the spongy mesophyll cells. And these organelles that are indicated by the letter D, they are the mitochondria. They would respire. They would also release the carbon dioxide gas, and that too would be utilized by these chloroplasts. So this is what they have mentioned in the statement of the question. The A that we have over here, this is the cell wall. The B that we have got here, so this is the large vacuole with us. The G letter is indicating the cytoplasm. The F letter that we have over here, this is indicating the cell membrane. The E over here is the chloroplast with us. The D that we have here is the mitochondria with us. And the C that we have over here, so this is the nucleus with us. But the thing he is asking us that we are giving you the scale bar, and this scale bar is equivalent to 0.01 millimeter, and they want you to convert this 0.01 millimeter into the micrometer. So you must know the conversion thing that 1 centimeter, this is equivalent to 10 millimeter. And whenever we have got 1 millimeter, so this is equivalent to 1000 micrometer. So if I have to go from the millimeter to the micrometer, then what I need to do is that this value that is 0.01, so I am required to be multiplying it with 1000. And when you would multiply it with 1000, so the answer that you would get here is 10 micrometer. Now I have explained everything that they are asking in the B, and they say that table 1.1 shows the function of some of the structures in the plant cell. And here you see they have given you the functions here. And this is some of the names of the structures where these functions would occur. So here they have given you the name of the structures where these functions are actually being accomplished. And then in the last, you could say, section of the table, they have given you the latter from figure 1.1. Now for the first structure they say is the nucleus, and as I have highlighted above, this is letter C with us. And if we have to write the function, so you could say is that this stores DNA, or the nucleus also makes messenger RNA. And the other interesting thing is that the ribosomes that we have got, they are also made in the nucleus. So you have to write just one part from here. They say chloroplast. The chloroplast letter is E with us, and the role of the chloroplast, you know, is to either photosynthesize or this would absorb the sunlight. So you have to write any one from here. They say function is aerobic respiration. We know The structure responsible for the aerobic respiration is mitochondria, and the latter that we are given for the mitochondria is D with us, so we would write D here. They say contains cell sap and stores water. So you see, this is the role of the large vacuole that we have here. So I have written large vacuole, and the letter that I have to be mentioning is this is B with us. They say A structure. So the A that we are given over here, so this is the cell wall, and we know the cell wall, it would maintain the structure of the plant cell. And this also prevents the bursting of the plant cells. So you can write any one from these. If we move on to word question number 12, they say is that we have got the red blood cells and they have the protein hemoglobin in them. And they are asking you to state the name of the four chemical elements. You don't have to be writing the symbols. You have to write the name of four chemical elements that are found in the protein. So we know that the proteins, they have carbon, they have hydrogen, they have oxygen. And in addition to these three elements, they also have got the nitrogen in them. For another 1 mark, they ask you to state the role of hemoglobin. So you would say that the hemoglobin that we have got within the red blood cells, this transports oxygen gas, or either you could say that they bind with oxygen gas. If we move toward the part B, and in the part B, they have given you a figure, and this is figure 2.1. And they have shown you the photomicrograph of the cells, and these cells, they are the red blood cells, and they have come from the person who is suffering with the sickle cell anemia. And we know that the red blood cells, they are the biconcave discs, and that biconcave discs is only possible if the person, they have got the normal allele for the hemoglobin protein, and that is HbA. But due to some mutations that the examiner is mentioning here, that some mutation has occurred for the gene that determines the hemoglobin protein, so that would be HbS. And when the allele is HbS, then the red blood cells, they happen to look like sickle shape. So these are the abnormal red blood cells. And if the allele is HbA, so the red blood cells, they would appear as the biconcave discs that we have got here. Now for the 2 marks, they are asking your suggestion that how the shape of abnormal red blood cell, which they have shown in figure 2.1, this would affect the blood flow. Now we know this fact that we have got the arteries. These arteries, they would divide into the arterioles. Then we would have got these miniature blood vessels. They are the blood capillaries. Then moving onward, we have the venules, and then here they would be coming up on the veins with us. At the level of the blood capillaries, we have got the cells, and these cells, they are in the need of the oxygen gas to be carrying out the aerobic respiration. So if I would have the normal red blood cell, which is a biconcave disc, so this biconcave disc, it would move smoothly within the blood capillaries. But due to the mutation that has happened, if the red blood cell would become a biconcave disc, so you could say is that they have got the tapering or the pointed ends, so they would get trapped here. And if they would get trapped here, then ultimately it would not only block the blood flow, but at the same time, this would enhance the clotting process. So for the 2 mark, you would say that if the red blood cells are sickle shaped, okay, and if they are not biconcave discs, so they would stuck in the blood capillaries, and by that they would increase the chance of blood clotting. And when these abnormal red blood cells, when they are bringing about a blockage in the blood capillaries, so for sure the flow of the blood would be minimized or this would be reduced. If I move you to question number 13, so they say is that we have given you figure 5.1. Here we have got 5.1, and here is a photomicrograph of the structure which is found in the animal and the plant cell. So whenever some structure happens to appear having this outer membrane, and if we have got the inner membrane that happens to look like this, so this is a confirmation that the organelle given is mitochondria. They say state the name and the function of the cell structure, which they have labeled it as A. So you would say this is mitochondria, and this would release energy by the respiration. Here you would score 2 marks. They say state the two pieces of the information that are needed to calculate the actual length of the cell structure which is labeled as A. Now you may have learned at some point that if we are required to be calculating the magnification, so this is equivalent to the image length, and we are required to be dividing it by the actual length. Okay, so this is the formula that we use to be calculating the magnification. So if we were required to be calculating the actual length, then the other two pieces of the information that we require is the magnification, and then we require the image length. So we would simply be moving the actual length here, the magnification would come here, and by that we would be able to be calculating the actual length. Now again for 1 mark, they have asked you to be interconverting the units. They say that the actual length of the cell structure A that they have shown you above, this is 0.0075 millimeter, and the examiner wants you to convert this value to the micrometer. And as I have mentioned above, that when you want to convert the millimeter into the micrometer, then what you are required to be doing is that the value which the examiner has given you, so you simply have to multiply that value by 1000. So if I have got this value and I would multiply it with 1000, then I would get the value in the micrometer. So I hope you would be clear with understanding the dimensions that come from this chapter number 2, which is organization of an organism. So here I have covered a lot of questions that have come in your recent past papers. So I hope this would be helpful. So see you in the next class for another topical past paper practice.